The second part of the morning talks begins with this one here. It's more about growth, more from the bioscience and biophysics viewpoint.
So thank you, Daryl. So what I will tell you about is mostly the work of Anna Maria Kiss, Eric Biot, Milan Cortizo, and Jasmine Burguet, in collaborations with the teams of Patrick Loos and Philippe André in Versailles near Paris. And well, time permitting, I will summarize in the end a piece of work that we did with mostly Karen Alim, Shahaf Armon, and in collaboration with Boris Schreiman. My main interest is about morphogenesis, how you go from a small one cell, a single cell, or a small group of cells to an organism that can be small or big in the animal world or in the plant world. So I'll work mostly on plants. And so the main question that I'm asking is what determines the trajectory in the space of shapes and what makes this trajectory robust to internal and external perturbations. So today I will speak about a part of our work that is more relevant to the topic of this workshop. So I am sorry I am venturing outside of my comfort zone, so please bear with me. And so what I will tell you about is a bit farther from what we do, because we combine biological experiments, physical measurements of the properties of tissues, and mechanistic models to try and address this question. So in the following I will focus on leaves. So in this here we're looking from the top at a small Arabidopsis plant. So the scale is here 1 centimeter. It's a weed that is used as a model system in plant developmental biology. And if we look at a single leaf that is a few less than a millimeter or a millimetric in size, well, that would be when it's say 8 days old, and it starts as a much smaller, a few, well, say 100 microns in size, and we want to understand how the leaf starts as a small bump at the tip of the plant and grows into a more elaborate shape with teeth that appear throughout time. Here this leaf has one pair of tooth that we guess here and there, and the shape is more complex at later times. And the leaf grows by a factor of about 1,000 in a few weeks. So what we will be interested in is how to obtain a trajectory in shape space from independent samples. So why work on leaves? So they're the main source of energy for life on this planet, for us as well, through photosynthesis. They have diverse shapes, so they can be simple with a tooth as Arabidopsis or without a tooth. They can be lobed as for an oak leaf, or they can be compound, composed of many small leaflets. So this is a single leaf, unlike what you could expect. And the leaflets can have teeth or not, and the structure can be even more compounds with leaflets on leaflets or lobes on leaflets, etc. So as a side remark, it's been observed that whether leaves are toothed or not, or untoothed, is correlated with mean annual temperature, and so sometimes the presence of tooth is used to infer information about paleoclimates. So here we want to analyze leaf shapes. There are many things that have been done previously. One of them was nicely explained by Chris in the first talk this morning. It's the use of landmarks to place homologous landmarks on leaves of the same species or of different species, and to look at how the landmark vary in the shapes, in the space of landmarks. Other approaches were based on discretizing the contours regularly, or the contours plus the midvein in this case, and doing principal component analysis for the discretized points. Other approaches.
At some point, don't you need to have more landmarks appearing during the evolution?
Yeah, when it's growing. So here, most of the previous work was done on mature leaves, in which you don't care about the fact that they grow, so you don't need to reparameterize or introduce new landmarks, etc.
But in principle, you would. You would need more landmarks. You could get more features.
Yeah, well, then you need, if you're using landmarks, you need to have a common set of landmarks across all the shapes that you're analyzing. So Chris would be better to answer this question, but so you need a common space in that case. Other researchers used decomposition in Fourier modes and the analysis of the Fourier coefficients. But as far as I know, there was little investigation of developmental trajectories, what happens if you look at leaves throughout times. So a big technical difficulty with leaves is that except in some specific cases, it's not possible to follow the same leaf throughout time from a very small state to a big state, because initially it's hidden by the older leaves. And so you would need to remove the older leaves, and that's perturbative. And so the question that the way we're addressing this is by sampling a big number of leaves at different developmental stages and trying to reconstruct a representative developmental trajectory from a large number of samples. So typically we collect a few hundreds of leaves at different stages, in a given genetic background, so for one type of plants. And this is a general strategy that we used, and I'll explain many of the steps in the following. So we image the leaf with a microscope. Here we're seeing chlorophyll fluorescence, and we can digitize this and obtain the contour of the leaf. From there, so there is some variability in where we cut the leaf when we isolate it from the mother plants. So we need to define by hand the limit of the boundary between the petiole and the blades, so the main part of the leaf. Then we automatically find landmarks. So the tip of the leaf, sinuses, and the tip of tooth. Sinuses are the creases in between teeth. So from there we can define a teeth hierarchy, and I'll explain you why we need this. So we could quantify things based only on the landmarks, and on the other hand, we reparameterize the contours using the landmarks as information, and we compute averages. And I'll explain the different steps in the following. So first, for the landmarks. So one technical difficulty when you have a large number of samples is to place landmarks by hand, which is often done in morphometric analysis. So we wanted to do something that is as automated as possible. So we define only the basis of the leaf, because this part is variable due to the position of cutting. The leaf tip is defined based on the greatest distance to the basis of the leaf. The sinuses are defined based on curvature, so there are extrema of curvature with the right sign. And the tooth tips can be defined based on curvature, so which is a very local criterion, or on symmetry, which is a less local criterion. It's basically whether in a given neighborhood of the point of interest, the part to the right is roughly symmetric to the part of to the left with respect to an axis. Well, to the normal, to the contour. So this allows us to identify the landmark, most of the landmarks automatically. And then if we think of, we'll need to define a certain level of homology between landmarks if we want to compare distances between landmarks between them. And one issue that arises, so if you look at this contour here, so you have one tooth, another tooth, another tooth, and another one. So it's the tip of the leaf would be here to the right. And so one question is, well, is this the first tooth? This is and then this the second, the third, etc. Or is there something else? So when we look throughout the development, we see that in general there's one tooth that appears close to the tip, and the second tooth here, and then the third tooth here, there. So this appears, seem to appear much later. So we need to know whether this is the second tooth or a secondary tooth. And in fact, if we think a little bit more about it, so there are primary teeth and secondary teeth, and in some species even tertiary teeth. So a more or less self-similar structure with teeth on teeth. And in this case, this is actually a secondary tooth. So we need to know that this landmark is not does not belong to the first tooth. So based on the angles between the teeth, so if this was the first and this was the second, this angle would be shallow. So based on the angles, we can define whether this is the teeth belong to the first hierarchy or to second order. So basically, we have a hierarchical algorithm to define the whether the teeth are primary or secondary, and we use the angles between the teeth, and we make successive tests, and from there we would know that this whole thing here is the first teeth, and this would be the first main sinus, the second main sinus.
So is there any sense of mechanism for the creation of the teeth? When I look at that upper right panel, it looks like a train of waves to me. Okay, so it looks like the leading wave is the biggest one and so forth.
So you'll see a movie with this afterwards. So there is indeed a wave, and we know many things on what defines the position of teeth. There is a patterning mechanism based on the transport of a hormone, auxin, and so we don't know it fully, but it seems that this is sufficient to prescribe teeth at regular positions, and the differences between teeth are due to the time of formation.
The labels in the C window does not correspond to the one in the window above, do they?
Okay, so maybe not.
Yeah, just order.
So it's 1, 2. They don't. Sorry. Yeah. Sorry for the confusion. No, they don't.
3 should go to.
3
Yeah.
should be 1.
Yeah, yeah. Sorry for that. Okay, so now from there we have the landmarks, and we can define homologous landmarks between leaves.
But in some situations, in fact, the landmarks are not really there. For instance, for the secondary.
Yes. So and what I will explain in the following, we use only the primary teeth. Well, the landmarks corresponding to the primary teeth, and even in some situations they're not there. So, and I'll explain how we deal with that afterwards.
So is there a fundamental shape associated with the landmarks? Like I'm thinking about this, the ones above with the red tips. Okay, when you think of them as waveforms, right? They seem like they're.
Well, the waveform changes throughout development. So all teeth are sharp. Young teeth are not as sharp. So yeah, you can't use. Well, you can define a local waveform. Now, if we want to to define average shapes, we need to define in a good way the average contours between the leaves. So this is somehow in the spirit of what Ian presented in the talk before. So we need to reparameterize the curves. So what Ian presented is the most evolved way to do it. So what we do is more basic. So we have a number of contours. The position, the vector as a function of the curvilineal coordinate, which varies between 0 and the maximal and the curvilinear length of the contour. And on each contour we have landmark. So the I corresponds to the contour index, and alpha to the landmark. So the landmarks can be, there is a maximum number of landmarks. So the first one is 0, and the last one is the maximum value, or the arc length, the total arc length. And some of the landmarks are absent in some contours. So not all of these are numbers. In order to have a common parametrization, so we transform the curvilinear coordinate as follows. So we define a mapping from the total length of the contour to 0, 1, that is piecewise affine, and such that the landmark number alpha is mapped to the rescaled average position of the landmark. So averaged over all contours. And if this landmark is not existing there, so it's not taken into account in the computation of the average. So the co-parametrized contours are the composition of this mapping and the vector field defined in the contour. So we have a common, after this, all contours are defined on a single interval, 0, 1, with all landmarks, when they exist, in correspondence to each other. So then we need to co-register the contours. So we minimize the distance between the contours, modulo rotations, translations, and scalings. So we minimize the L2 distance between all contours. So the sum over I different from J of the difference between the two, with R, big R rotation translations, and rho scalings. And so this can be solved iteratively. And so the outcome is the registered contours with the corresponding landmarks. So we put the rotations and we remove the scaling factors, because we want to keep track of of size information. So we have a common parameterization for all contours with possible variations in size between contours. So here's what we... Yeah?
I didn't understand how you decide whether a landmark is valid or not. How do you recognize it automatically?
So the landmarks are recognized based on, well, to simplify curvature. There's a certain number of landmarks, and so they're numbered according to their position along the contour. So we know the type of landmarks, whether it's a sinus or a tooth, tip, etc. So we associate the landmarks in we use a hypothesis that the oldest leaf, or leaf number 1, is the closest to the tip. So maybe that was the missing information. So now after that we average. First we look mostly at the average. We average all the contours, and so this is a comparison of what we would get, in yellow here, if we didn't use landmarks. So the leaf, if we use landmarks to register, the leaf tip is, the average contour is more representative of actual leaf tips. If we look at teeth, the tooth tip is slightly more representative of actual leaves, and the more we add landmarks, the more the contour is representative of actual leaf, because if there is variability in the position of landmarks, as Ian explained, if you average brutally, you lose the information on the shape of the teeth. And so using this representation allows to get a representative shape for leaf tip. So we applied it to various species, and in many cases, if you average, so the gray contours here are what you get without taking into account landmarks. If you take the landmarks into account, you get more representative averages. And this is true also for hands, so that was as a fun exercise. And now how to get developmental trajectories. So by looking at a large number of samples, we can look at how length changes with age of the plant. So this would be these are trajectories for different leaves. So for instance, if we look at the first leaf, it appears the first, it grows in size and then saturates at some point at say 38 days, and same for the other leaves. So there is on average a mapping between length of the leaf and the plant age. So we use length as a proxy of age. And then what we do is, we define the average shape for each length, or for a small range of for leaves in a small range of lengths around 1, and we can do a sliding average to define the average leaf shape for a certain range of lengths. So this is a movie for what we get for the average trajectory. So initially you see the teeth appearing, so you get to the maximum size, and after some point the shape changes globally with the same number of teeth. So the first tooth are the closest to the tip, and the second and third appear later.
Yes, so we have the impression that here that you have creation of new coordinates
near the basis. Yes,
near the basis, because you have.
That's right.
Not just an impression, right? I mean, there is creation.
Well, there is creation near the basis of the leaf. So if we look at how the leaf grows, so there's more. It grows more at the basis than at the tip. At least during certain intervals of time.
Also expanding, so it's growing in the interior.
Yeah, it's growing all around. And so this is, what you were saying is here is clearly seen here. So this is the rescaled shape of the leaf, of a young leaf and an older leaf. And the what is drawn is the position of successive sinuses according to age. So older leaves have the teeth closer to the tip. And so one surprising thing was that some of the features, like this first tooth, disappear for at later stages.
So you have some kind of cluster algorithm to detect the homology between your primary tooth?
To detect
But the question of Laurent, I guess, like, or you have the tree structure. How do you identify when a primary tooth disappears at the end or doesn't exist in the beginning?
so we that's true. So we had for that we did it by hand. For the appearance, there's no problem because we use the distance. We use first the distance from the tip to define whether it's the first, second, etc. So this criterion fails for the later stages, and we had to look big. When first we didn't know that, so we saw discontinuities in the data. So and then we reconsidered the redefinition of the landmarks. So we needed to state formally that this is the second tooth.
So you do that by hand?
Yes, we ended up doing that by hand. That's right. Okay, so well, I've prepared a little bit too much material. This allows us to study the mechanism that control leaf growth. So we can compare two genotypes between them. For instance, a genotype in which there is no tooth, well, which leads to the green contour here, and a genotype in which there are teeth, and see that initially teeth appear, well, this suggests that initially teeth appear through a reduced growth at the sinuses. At later stages, it's reduced growth at the sinuses and more growth at the tips. So just now I'll try to summarize the few slides in one sentence. So the contour, it's a nice information, but it's only part of the information. And we're also interested in what happens in the blade of the leaf. And so one thing that was inspired by Darcy Thompson is the use of conformal maps. So conformal map, one nice property of conformal maps in 2D is that if you know the contour, you know the transformation everywhere. So what we did, we looked at growing leaves. So these are older leaves from another species, Petunia. We digitized the contours, and at later stages, we can follow their growth. So we can follow the contours plus 3 landmarks, define the conformal map from the contour and the landmarks, and then compare the conformal map to the actual growth. So we find that there is a very good agreement for displacements between the conformal map and the actual measured displacement within the leaf, but not so for the growth rates. There are derivatives of displacement, so there are small-scale fluctuations that we don't capture. So we capture the big trends in the growth of the inside of the leaf, but not the small-scale features. So, and I'll stop here. So by combining landmarks and contours, we can analyze the developmental trajectory of a leaf. And contours give all information for 2D isotopically growing systems, because that's what corresponds to the condition of conformality. Thank you for your attention.
Well, we have time for a few questions. Does anyone have any questions? Yes.
Does it make sense, instead of having landmarks, to say that you think in terms of coordinates And you know, during the development, you instantiate all the events in this kind of coordinate.
So you
I think you are doing it in the other way. You start from the landmark and create the coordinate, but you could.
yes, you could.
Does it make sense for you?
It does. Well, to reinitiate the coordinates, but you need to know how to do it. So the use of the landmarks are the only points that we know. Well, their homology. So we know for sure that one landmark is mapped on the same landmark throughout growth. So that's a very strong information. If you were to reinitiate coordinates, you would need to have a model for how growth is occurring and then compare it to what you get.
Yeah, because in the full development, if you could have some kind of grid, what you will see something very. You will see new coordinates appearing at the base, or you will see just some huge spacing.
It's not only at the base; they appear on all the length of the leaf,
No,
and there is a gradient.
dilation, I think you could have dilation. You could have just dilation. And
No, dilation is
also
not.
something which
It's
cannot
not
be
dilation
treated
is not sufficient. So it's the growth is really inhomogeneous.
Yeah.
Ah, you mean the you have local dilation if you want, but there are strong variations in the dilation along the contour, and so the use of landmarks allows us to have to constrain the dilations on some of the intervals along the contour.
If we were looking at one of the landmarks which you see, then we wouldn't always be looking at the same piece of material, right? Well, material will be flowing through those.
No material is not flowing through those. So that's what we know from
so
different sets of,
same material.
observation, is that if it was the same leaf, it would be the same material at the landmark. So there are material points, these landmarks.
You showed leaf 7. Do all leaf 1, 2, or the other one, do they have the same shape
No.
up to dilation of different development?
No, they don't.
What are the
They don't.
findings?
So there are variations according to leaf rank. So so let's say up to well, it depends on the growth conditions first. But there's a certain range of leaves that are juvenile leaves that do not have hair or do not have hair on the two sides of the leaves, and they behave a bit differently from adult leaves and from later leaves. So people tend to define 3 stages. So we roughly find them in the trajectories, but the stages are not sharp cuts.
You can't find the stages from your analysis.
So we could. So if we had the criterion, if we try to define stages in a rigorous way, we could extract stages from this analysis.
Couple more questions. Yeah, go ahead.
So your specific method only works for leaves that have teeth. And can you generalize it to things like blade of grass that doesn't have teeth?
So it works if you have enough landmarks. So for blades of grass, it wouldn't, or it would do rather poorly because, well, there's a landmark at the tip. Well, that's well defined, but there is, for grass, there is a strong gradient in growth rate, so it's growing more at the basis, so like most leaves, and so you would miss that. But in the case of grass, the shape is well, they have slowly varying width, so the shape there's less to learn about the shape.
Easier. Yeah. So Jane.
Did you say, so coming back to the conformal mapping part, you showed in the middle, they seem to be given by a conformal map. But suppose you include the whole leaf, including the edges, are they also?
No, it's not. It's a whole. It's a whole
The
leaf.
whole leaf is conformal.
Yes. Well, I showed the grid only on part, but just as an illustration. But it's from the contour
Yeah.
to the whole leaf.
So once you have the conformal map, f of z, let's say, and the modulus of f prime of z, that's the growth rate.
That's right.
And so then you can plot that, and I wonder, what does it look like for different kinds of leaves? Whether they, have the growth rate higher at the roots.
So that's that was I went fast. That's what we get. So that's the modulus of f prime. So they're going. So here the basis of the leaf is here. Here the basis is there. So there's more growth at the basis and less at the tip.
And moreover, those growth, the fast growth might correspond to your teeth. Is that true?
Yeah. So these species do not have teeth.
They
So-
don't have teeth. Okay.
Okay. So we didn't.
Other cases. Yeah.
Yeah. So we didn't. Well, it happened that for historical reasons this was done way before, and we tried only leaves with smooth contours at that time.
Are we satisfied? Thank you.